Today we're going to take a look at how DNA replicates. So I'll start at the top by drawing a simple sketch of a double helix. Those are two strands of DNA, the backbones, held together by hydrogen bonds. So here I'm representing 10 different base pairs. Each set of hydrogen bonds, that would either be 2 hydrogen bonds between A's and T's or 3 between G's and C's. I'm just representing with a single vertical line here, showing the two strands being held together. And what we're going to focus on today are 2 intermediate stages that I'm not going to show right now. The goal, the outcome of this process, is to have 2 identical double helices or chromosomes after DNA replication is complete. So in between the start and the stop, I'm going to show 2 different intermediate stages. One is referred to as the replication bubble, where DNA replication begins. And this is a region of a double helix where the hydrogen bonds holding the 2 strands together are broken so that we have single non-hydrogen bonded nucleotides that are in that bubble. And the rest of the double helix is still hydrogen bonded. And later in the process, that replication bubble, as we'll see, expands. So right before replication is complete, our replication bubble might look like this, where there might be a single hydrogen bond or set of hydrogen bonds holding 2 nucleotides together on either end of our piece of chromosome that's being replicated. So again, here we're going to look at DNA replication over time, starting at the top with one double helix, moving through time to the replication bubble opening. Farther in time, the replication bubble gets larger, and eventually those separate into 2 different double helices that are identical to each other. So now we need to focus a little bit on the details. The first thing I'll do is actually give our DNA a name, so to speak, by adding in some nucleotides so that we see the actual nucleotide sequence of these pieces of DNA. So here on the top strand, I've just written in some nucleotides, and you'll notice, as is typical for DNA, that the 2 strands are complementary to each other, meaning that A's are base paired with T's, T's with A's, G's with C's, and C's with G's. So we have a complete double helix here. And now what I'm going to do, because we need to know a little bit of something about the polarity of the DNA, is I'm going to add in the 5' and 3' notations. Arbitrarily, I'll say the top strand is from 5' on the left to 3' on the right. And because DNA strands are anti-parallel, we'll make the bottom strand then 5' on the right and 3' on the left. So we can continue those nucleotides and polarities through the rest of this series of images, so I'll add those in now. So here I've added all of the polarities and the original, this is a critical point here, the original 2 strands of DNA, the horizontal white lines, and their nucleotide sequences. And you'll notice that down here at the very last step at the bottom of the slide, that I've only written in 2 strands of nucleotide sequence. These are the original 2 strands, the top strand from 5' on the left to 3' on the right, shown up here at the top, and also the original bottom strand, the one that reads 5' on the right to 3' on the left, is down here. And you'll notice that these 2 original strands are part of separate chromosomes after DNA replication, and this is why DNA replication is referred to as semi-conservative, meaning the 2 daughter double helices, daughter chromosomes, that are produced by replication, half of the nucleotides, or one of the 2 strands, is the original strand, one of the original strands. The second strand of each chromosome produced by replication is new DNA that's synthesized as part of this process. So let me provide a little color coding key here in the upper right-hand corner. The white strands represent DNA, and I'm going to add some blue nucleotides, and that's going to indicate RNA, which may seem a little bit strange, but this is how our cells do DNA replication. So now let's take a look at what's happening inside this replication bubble. As soon as our cells have the origin of replication, which is where this replication bubble forms, when it melts or denatures, which means that the hydrogen bonds have broken, as I've shown here, the first thing that happens during DNA replication is a piece of RNA, not DNA, but ribonucleic acid, is synthesized. And it's a short piece of RNA, not necessarily 2 base pairs, as I'm showing here, typically more like 20 perhaps. And the RNA then is base pairing with the top strand of DNA, which means that its nucleotide sequence would be C pairs with G and A pairs with T. So right now we have a DNA-RNA hybrid molecule. It's base paired here. The blue strand is RNA, the white strand is our original DNA chromosome. And because this RNA strand is base paired with the top DNA strand, which is 5' on the left, we also know the polarity of this RNA strand, which means it has to have its 5' end on the right, and therefore its 3' end on the left, that A. So again, our cells synthesize these RNA primers specifically to bind with the DNA in the replication bubble. And the same thing happens on the bottom strand. So for example, an RNA primer might be synthesized here to base pair with the G and the T. And in this case, by chance, it happens that the primers have the same sequence. There's the 5' end, there's a C, and then base pairing with a T, there's an A. And that's the 3' end of that RNA primer, the blue molecule. 5' C, 3' A. Now is where DNA replication starts. And what happens now is the enzyme, which is a protein, DNA polymerase, represented by this circle, will come in and bind to the 3' end of each of the 2 primers and add in the next nucleotide that's complementary to the DNA strand. So there'd be a polymerase molecule at that 3' end of the primer, and there'd be another polymerase that's bound to the 3' end of the primer that's base paired with the top DNA strand. And so what happens next is DNA polymerase, this enzyme, adds in the correct DNA nucleotides, like a T will base pair with that A, and a T with the A on the bottom strand. This is the actual step of DNA replication, where DNA polymerase, the orange circles representing these DNA polymerase enzymes, start at the primer, the 3' end, and add in each consecutive nucleotide that's base pairing with the top template strand here, or the bottom template strand down here. And now the 2 polymerase proteins move to the next nucleotide that needs to be synthesized. In other words, we have these 2 nucleotide RNA primers in blue, and now they're ending with a DNA nucleotide, the T on both strands. So now it's the 3' nucleotide. So DNA polymerase now adds a new nucleotide onto the 3' ends of these growing strands of nucleic acid, and C base pairs with G, and C base pairs with G. So this is how DNA replication works, with DNA polymerase adding a new nucleotide to the end of each growing strand, and that new nucleotide is complementary to whatever is the nucleotide on the template strand. So here we have a C, that's why DNA polymerase added a G. Now if we look sometime into the future, this is what our enzymes are going to have done to these 2 template strands. And now, somewhat later in time, our DNA polymerase enzymes have reached where the chromosome is still hydrogen bonded at each side. So these polymerase molecules have moved along the template strand, adding in these new nucleotides, DNA nucleotides, in white, forming new double helices. Again, these started with the primers in blue, and the polymerase molecule has been moving in each direction towards what we call the replication fork. And this is where that double-stranded piece of DNA splits into two single strands, sort of like a two-tined fork. So these replication forks are the edges of the replication bubble. Now at this point, there are two new pieces of vocabulary to add in. These are the leading strands. So in the top left and in the bottom right, we have leading strands of DNA. This strand that starts at the primer that was at the origin of replication originally and moves towards one of the replication forks, and the strand that's newly synthesized that's complementary to the bottom strand of DNA, the original template strand. So these two are the leading strands. Now we've got a problem here because with just these two molecules of DNA polymerase, we're only synthesizing DNA that's complementary to the top strand on the left and the bottom strand on the right, the leading strands. But how are we going to get to where we're trying to get at the bottom of the slide here, where we have the entire top template strand synthesized? In other words, and also, I should say, the bottom template strand needs to be fully synthesized. So we have two nucleotides that are still single-stranded. So the question is, how do those get replicated? And this is where the lagging strands, also known as Okazaki fragments, come in. And these are referred to as lagging because their synthesis starts later than the leading strand synthesis. Note that earlier in time, these nucleotides, the RNA primers were added and new DNA nucleotides in white were being added in. That's early. Now that we have some unreplicated nucleotides that are in the replication bubble, what happens is new RNA primers would be synthesized. So in this case, because I'm running out of nucleotides, I'll just suggest maybe there's a one nucleotide RNA primer there in the upper right, an A. And the same would be true down here on the bottom template strand, a new RNA. And in this case, this is RNA in blue. So complementary to A in DNA is T in DNA, but remember there's no thymine in RNA. Instead, there's uracil, U. So here we have two new primers that have been synthesized and are base pairing with our template strands, and those have two new 3' ends. And so we can have more of those molecules of DNA polymerase grab those 3' ends. I'm not going to show the RNA polymerase in this case, and synthesize more DNA. So complementary to a C in the upper right would be G, and complementary to C in the lower left is also G. So now we're at the position where we have almost fully synthesized this entire chromosome. The last thing that would happen, which I'm not showing here, is that the hydrogen bonds holding together the terminal A's and T's would break, and that DNA polymerase molecule would continue and synthesize that last nucleotide on both the top and the bottom template strands. So to reiterate, we have leading strands. Those are the strands that started with the original primers up here at the start of the origin of replication and the start of replication. Those are the leading strands. DNA polymerase will continue reading the top template strand until polymerase literally slides off the telomere, the end of the chromosome, and then replication is complete. And the same thing would be true on the bottom strand here. That polymerase molecule would read that T, add an A, and that would be the end of the template, so synthesis would be complete at that point. There's an issue here then, which is that there are a couple of regions of the chromosome that don't get replicated. For example, in this case, we have, looking at the lower left side, on the bottom strand, we have this RNA primer in blue, a U, and that G was synthesized and is covalently attached to the U. So we have a strand of RNA DNA hybrid molecule there. But how are we going to get a nucleotide that base pairs with this lower left T at the 3' end of the template strand? Well, we would need a primer, but there's no place to synthesize a primer out here because there's no template left. So in this scenario, the T that I'm circling in red and the A that I'm circling in the upper right-hand corner, the 3' ends of the 2 template strands will not be synthesized. This is called the end replication issue, and we can talk about this more in class or in a future video. So this is one of the drawbacks, or one of the reasons why DNA replication isn't perfect. A little bit of each end of the chromosome doesn't get synthesized or replicated at the end of DNA replication. There are a couple of other issues to mention that I'm not going to go into detail about now, but we'll either talk about this in class or I'll add it in a future video. When we look at the newly synthesized DNA that's in the interior here of this replication bubble, this, for example, on the top strand, there's a C and then an A. Those are covalently bound to each other on the backbone, and that A is covalently bound to the T, to the G, to the C, to the A. And this U and this G, the second primer and its DNA nucleotide that was added onto its 3' end, are also covalently bound. But here in yellow, circled, there's a break in the DNA sugar phosphate backbone. That G and that C are not covalently bound to each other. This is something else that our cells take care of as soon as DNA replication finishes before the cell cycle proceeds. Likewise, in between these two pieces of newly synthesized nucleic acid, there's also a gap in the sugar phosphate backbone of the nucleotide sequences. Those get corrected later. The last issue that has to be dealt with, which we're not going to go into detail, is everything in blue, all of these nucleotides are RNA, but we don't want RNA in our DNA chromosomes. So at some point near the very end of replication, a special DNA polymerase comes in and removes all of the RNA molecules and replaces them with DNA. So to conclude, now we can actually read the DNA sequences of this newly synthesized nucleic acid and add those to the picture down here at the bottom when replication is finished. And as I said, there are still some RNA nucleotides in here. Those would be replaced later with DNA nucleotides. And as I mentioned, that eventually this molecule of DNA polymerase on the left would read that A and add in a T. So the top nucleotide sequence was template. Again, that's one of the original DNA strands that we started with, and so is the very bottom strand. That's template DNA. Those are the 2 nucleotide sequences we started with at the beginning of this process. The 2 in the middle here are the ones that were just newly synthesized, and as I noted, there's no place on the upper right here to synthesize a primer out here floating in space that could be used to add in whatever is complementary to this A, and that's why there is no nucleotide there on the very end of this chromosome. Likewise, on the bottom chromosome, I shouldn't have drawn in that last nucleotide because there's nothing that's been synthesized here complementary to this T. There's no place to put a primer out here hanging out in space to allow DNA polymerase to synthesize an A there. But we can add in the rest of the nucleotides. We had U complementary with A, and then it was used to synthesize this G. And we had the original primer sequence, C with A, that was used to synthesize T, G, C, T, and then an A out here after this polymerase enzyme reads that last T and adds in the complementary nucleotide. So notice that now we've gone from one double helix, whose top strand read ATG, CATG, CTA, to 2 copies of double helix. We've got the template strand, the top strand here, and its complementary strand. And we've got the original template strand and the top strand that was synthesized complementary to it, And so when you read the top strands, they are TGCATGCTA, U, which will be replaced with TGCATGCTA. So now we have the two top strands are identical to each other. The two bottom strands are identical to each other. We have successfully synthesized, replicated. One double helix has become two new double helices that are identical in sequence. That is DNA replication.